Morning students, we are discussing factors affecting enzyme activity. What? Factors affecting enzyme activity. In yesterday's session, in yesterday's class, right, we discussed about a substrate concentration. Right? So what is the enzyme that dissolve the bacterial cell wall? Lysozyme. The enzyme that dissolves bacterial cell wall is a lysozyme. For lysozyme enzyme, turnover number is 30. For lysozyme enzyme, turnover number is how much, sir? 30. It is having the least turnover number. Who, sir? Lysozyme. What is its turnover number? 30. Okay, right. Now, go for the next factor that affects enzyme activity, that is enzyme inhibitors. What, sir? Enzyme inhibitors. Enzyme inhibitors. Now, listen here. Sir, these enzyme inhibitors, these are chemicals. When these chemicals, they binds to the enzyme, these chemicals, they will inhibit enzyme activity. Such chemicals are called as enzyme inhibitors. Therefore, enzyme inhibitors are chemicals. What they are doing? When they binds to the enzyme, they inhibit the enzyme activity. What they are inhibiting? Enzyme activity they are inhibiting when they binds to the enzyme. Such chemicals are called as enzyme inhibitors. Okay, therefore enzyme inhibitors are chemicals which when binds to an enzyme, what they do, sir? They shut off or they shuts off or inhibits what? Enzyme activity. These chemicals, when they binds to an enzyme and inhibiting the enzyme activity, these chemicals are called as enzyme inhibitors. What are these chemicals called as, enzyme inhibitors? Okay, the mechanism by which a enzyme inhibitor inhibits the enzyme activity, that mechanism is called as enzyme inhibition. Mechanism by which a enzyme inhibitor inhibits the enzyme activity called as what, sir? What it is called as? Called as enzyme inhibition. Mechanism by which right, an enzyme inhibitor, I'm speaking about mechanism. Okay, right, therefore mechanism by which right, an enzyme inhibitor inhibits enzyme activity. What is that mechanism called as? That mechanism is called as enzyme inhibition. What is that mechanism called as? Enzyme inhibition. Sir, enzyme inhibition in general of 3 types. In general, this enzyme inhibition is of how many types? 3 types. What are they, sir? Competitive inhibition, competitive reversible inhibition, non-competitive irreversible inhibition, non-competitive reversible inhibition. Therefore, it's of how many types? 3 types. What are the 3 types, sir? First one, number 1, competitive reversible inhibition. Second one is non-competitive non-competitive irreversible. Non-competitive irreversible inhibition. Non-competitive irreversible inhibition. And third one is a non-competitive reversible. Non-competitive reversible inhibition. Non-competitive reversible inhibition. Okay, shall we go for the first one? What it is? Competitive reversible inhibition, right? Go for the first one. What it is? Competitive reversible inhibition. Competitive reversible inhibition. See this diagram once. Sir, this is enzyme, sir. What is this one, sir? This is a enzyme. This is a enzyme. Okay. This is active site. What is this one, sir? This is a active site. This is a active site. This is the substrate which has to bind in the active site of this enzyme. Substrate which has to bind in the active site of this enzyme. Sir, this is competitive inhibitor. What is this one, sir? Competitive inhibitor. See this ones. Competitive inhibitor is structurally similar to substrate molecule. Competitive inhibitor is structurally similar to substrate molecule. So we are calling competitive inhibitor as a structural analog or substrate analog. Better to call substrate analog because it is a structurally similar to substrate molecule. So competitive inhibitor is a substrate analog. Now you tell me, sir, this competitive inhibitor is structurally similar to substrate molecule. So competitive inhibitor competes with substrate molecule. Competitive inhibitor competes with substrate molecule. And instead of substrate, competitive inhibitor goes and binds in the active site of the enzyme. Once a competitive inhibitor binds in the active site of an enzyme, what has binded in the active site of the enzymes are competitive inhibitor. Once a competitive inhibitor binds in the active site of an enzyme, right, the enzyme activity is inhibited. Enzyme activity is inhibited. What is inhibited? Enzyme activity is inhibited. Sir, why we are calling it as a competitive inhibition, sir? Because this is competing with substrate molecule and inhibiting the activity of enzyme. So, this competitive inhibitor competitively inhibits the enzyme activity. Should be very careful. It is competitively inhibiting the enzyme activity. Competitively inhibiting the enzyme activity. Is this statement okay? Right? I am writing these statements on the board. Right? Sir, competitive inhibitor, CI stands for competitive inhibitor. Competitive inhibitor is it is structurally similar. Structurally similar to what? Substrate molecule. Structurally similar to substrate molecule. Structural similarity. Okay, molecular structure I am writing. Molecular structure. It means the molecular structure of competitive inhibitor is similar to that of what, sir? Substrate molecule. So we are calling competitive inhibitor as what, sir? So competitive inhibitor is a substrate. So it is a substrate analog. Okay, who is a substrate analog, sir? Competitive inhibitor is a substrate analog. Sir, what this competitive inhibitor is doing, sir? Competitive inhibitor competes with what? Competes with substrate. Competes with substrate molecule. Competes with substrate molecule. Okay, competitive inhibitor, what it does? Competitive inhibitor binds where? Binds in the active site of the enzyme. Should be very careful. At where a competitive inhibitor is binding, competitive inhibitor binds in the active site of an enzyme. Active site of an enzyme. Sir, once competitive inhibitor binds in the active site of an enzyme, enzyme gets inhibited. Yes or no? Right. I am writing the statement here. Once what? Once competitive inhibitor binds where? In the active site. Once competitive inhibitor binds in the active site of an enzyme, active site of an enzyme, right? Enzyme gets inhibited. What gets inhibited, sir? Enzyme gets inhibited when competitive inhibitor binds at active site of the enzyme. The enzyme gets inhibited. Therefore, competitive inhibitor, what, sir? Competitive inhibitor competitively, what, sir? Competitively inhibiting what? Enzyme activity. It is inhibiting substrate or it is inhibiting the enzyme activity? It is inhibiting the enzyme activity, competitively inhibiting the enzyme activity. Okay, right? Sir, see this diagram once. Sir, here what and what are there, sir? This is enzyme. This is competitive inhibitor. Yes or no? This is enzyme. This is competitive inhibitor. Sir, actually, who should bind in the active site of the enzyme? Substrate. If a substrate binds in the active site of the enzyme, then what is formed? Enzyme substrate complex. But as a competitive inhibitor is binded in the active site of the enzyme, here enzyme substrate complex, is it formed? Not formed, no. What is not formed? Enzyme substrate complex is not formed. Enzyme substrate complex is not formed. Okay. Sir, why we are calling it as a reversible, sir? Why we are calling it as a reversible? Because this competitive inhibition can be reversed. Competitive inhibition can be reversed. How, sir? By increasing the substrate concentration. If substrate concentration is more, then the substrate binds in the active site. Substrate binds in the active site of the enzyme. It won't allows the competitive inhibitor to bind, because this is purely competitive. Binding of substrate to what, sir? Active site of an enzyme is purely competitive. It competes. Is this statement okay? Right? Therefore, right? Competitive inhibition can be reversible. Competitive what? Inhibition can be, competitive inhibition can be reversible. Can be reversible. How, sir? By increasing what? Substrate concentration. By increasing substrate concentration, right? This competitive inhibition, we can reverse it. Therefore, competitive inhibition is reversible by increasing the what, sir? Substrate concentration. Okay, shall we go for the examples, right? Now go for examples of competitive reversible inhibition, right? Now here I am writing one equation. Here I am writing one equation. See here. Succinic acid. Succinic acid, what it is, sir? Succinic acid plus FAD plus gives rise to what, sir? Fumaric acid. What is formed, sir? Fumaric acid. Fumaric acid is formed. Along with fumaric acid, what is formed, sir? FADH2 is formed. What is formed, sir? FADH2 is formed. Sir, this is one of the reaction in Krebs cycle. Strictly speaking, right, this is oxidation reaction of Krebs cycle. This is oxidation reaction of what, sir? Krebs cycle. This is oxidation. This is what, sir? Oxidation reaction. This is oxidation reaction of what? Krebs cycle. This is oxidation reaction of Krebs cycle. Sir, this reaction is catalyzed by one enzyme by name succinic dehydrogenase. What is the enzyme name, sir? Succinic dehydrogenase. This succinic dehydrogenase is called as a marker enzyme. What is this succinic dehydrogenase enzyme called as a marker enzyme? Marker enzyme. Sir, in this reaction, what is the substrate, sir? Succinic acid. This is the substrate. Yes or no? In this reaction, I am speaking about this reaction. In this reaction, succinic acid is a substrate, fumaric acid is the product. Fumaric acid is the product. What is the enzyme involved here? Succinic dehydrogenase, right? Now listen the statements. Malonic acid. Malonic acid. If it is present in the form of a ion, we will call it as a malonate. Ion means malonate, compound means malonic acid. Okay, right? Now, sir, what is this compound name, sir? Malonic acid. Sir, this malonic acid is a competitive inhibitor. Malonic acid is a competitive inhibitor. Sir, this malonic acid is structurally similar to succinic acid. Malonic acid, how it is? Structurally similar. It is structurally similar to what, sir? It is structurally similar to succinic acid. Structurally similar to succinic acid. See here, malonic acid is a competitive inhibitor. Succinic acid is a substrate. Sir, what is similar to what, sir? Malonic acid is structurally similar to succinic acid. So, malonic acid competes with succinic acid. What competes with what, sir? Malonic acid. Malonic acid competes. Malonic acid competes with what, sir? Succinic acid. Malonic acid competes with succinic acid. Okay, what is competing with what? Malonic acid competes with succinic acid, and malonic acid goes and binds in the active site of the enzyme. Malonic acid binds in the active site of the enzyme. Which enzyme? Succinic dehydrogenase enzyme. So the enzyme gets inhibited. When malonic acid what? Binds where? Binds in the active site. Once malonic acid binds in the active site, okay, of which enzyme, sir? Succinic dehydrogenase enzyme. Succinic dehydrogenase enzyme. Then what happens? The enzyme gets, what happens? The enzyme gets inhibited. The enzyme gets inhibited. So, malonic acid is a competitive inhibitor of succinic dehydrogenase enzyme. What it is, sir? Malonic acid is a competitive inhibitor of what, sir? Succinic dehydrogenase enzyme. Right? Okay, right. Therefore, what is it, sir? Malonic acid. Malonic acid is a what? Malonic acid is a competitive inhibitor. Competitive inhibitor of what? Should be careful, I am not writing the substrate name, I am writing the enzyme name. Succinic dehydrogenase enzyme. This statement is important, should be careful with this statement. Therefore, malonic acid, it is a competitive inhibitor of succinic dehydrogenase enzyme. With this, we completed the first example for competitive reversible inhibition. Now go for the second example. Okay, second example. See here. Sir, competitive inhibitors. We are discussing examples for competitive inhibitors. Example is sulfa drugs. What, sir? Sulfa drugs. How, sir? Sir, competitive inhibitors, what, sir? Competitive inhibitors are often used often used to control bacterial pathogens. This statement is important. Sir, how competitive inhibitors are used to control bacterial pathogens, sir? Right? See here. Here I am writing one equation. PABA. P-A-B-A. Sir, what is the full form of PABA, sir? P-amino benzoic acid. What it is? P-amino benzoic acid. P-amino benzoic acid. Sir, this PABA, what sir? It gets converted into folic acid. Gets converted into what, sir? Folic acid. Folic acid. In presence of an enzyme by name, what, sir? DHPS. What is the enzyme name, sir? DHPS enzyme. DHPS enzyme. Sir, in this reaction, this PABA is substrate. PABA is what, sir? Substrate. Whereas folic acid is the product. Folic acid is what, sir? Product. See here. Sir, here what is the substrate, sir? PABA. It gets converted into what? Folic acid. This folic acid is required for bacteria. Folic acid is required for bacteria. If a bacteria requires folic acid, right, then how it gets that folic acid? By this reaction. Okay. In this reaction, what is the substrate, sir? PABA. Sir, sulfa drugs. Sulfa drugs, they are structurally similar to PABA. So sulfa drugs competes with PABA. So sulfa drugs binds in the active site of DHPS enzyme. When sulfa drugs binds in the DHPS enzyme, this enzyme gets inhibited. Once enzyme is inhibited, PABA is not converted into folic acid. If folic acid is not formed, bacteria cannot survive. Therefore, sulfa drugs are used to control bacterial pathogens, but sulfa drug is a competitive inhibitor. So, competitive inhibitors are often used to control bacterial pathogens. Yes or no? Right. Sir, sulfa drugs, they are structurally similar to PABA. So, sulfa drugs competes with PABA and sulfa drugs binds in the active site of DHPS enzyme. So the enzyme gets inhibited. Who inhibited this enzyme? Sulfa drugs. They are competitive inhibitors of this enzyme because competing with PABA and inhibiting the enzyme. Competing with PABA, inhibiting the enzyme. So sulfa drugs are competitive inhibitors of DHPS enzyme by which this enzyme gets inhibited. No? So substrate not converted into product. If this folic acid is not formed, bacteria cannot survive. Therefore, sulfa drugs, competitive inhibitors, are often used to control bacterial pathogens. Is this statement okay? Right? I am writing here one important statement. Right, sir? What it is? Sulfa drugs are structurally similar. Structurally, what, sir? Similar. Structurally similar to what, sir? PABA. Structurally similar to PABA. Okay. So what happens, sir? Sulfa drugs. What? Sulfa drugs competes with what, sir? PABA. And what happens? Binds where? Binds in the active site of what? Enzyme. What is that enzyme name? DHPS. DHPS enzyme. So what happened? Sulfa drugs are competitive inhibitors. They competitively inhibits which enzymes are? DHPS enzyme. Okay. Sulfa drugs are competitive inhibitors. This sulfa drugs competitively inhibits Sulfa drugs competitively inhibits what, sir? Competitively inhibits which enzyme? DHPS enzyme, right? Go for the second one. What it is? Non-competitive irreversible inhibition. What it is? Non-competitive irreversible inhibition. Non-competitive irreversible inhibition. Sir, this is a enzyme. This is enzyme. Sir, this is active site. What it is? It is a active site. It's active site. Sir, what is this one, sir? This is not a active site. This is a other site. This is not active, other than active site. Other than active site. Okay, right. Sir, this is a non-competitive inhibitor. Non-competitive inhibitor. This is a substrate. Now see this diagram. Non-competitive inhibitor. It is not structurally similar to substrate. So, non-competitive inhibitor does not compete with substrate. Non-competitive inhibitor goes and binds right at other site but not in the active site. Once this non-competitive inhibitor binds at other site of an enzyme, SH groups of enzyme are destroyed. SH groups of enzyme are destroyed. Once SH groups of enzymes are destroyed, okay, the globular structure of the enzyme changes. Globular structure of the enzyme changes. Due to change in globular structure of the enzyme, right, the shape of the active site also changes. Shape of active site changes. Sir, how the shape of this active site changes, sir? Right, the active site, right, it becomes like this. Now, can a substrate comes and binds? No. Therefore, enzyme gets inhibited. What gets inhibited? Enzyme gets inhibited. Sir, here enzyme substrate complex, is it formed? No. Substrate has not yet binded. Why substrate has not binded? Because the shape of active site has changed. Why shape of active site has changed? Because globular structure of enzyme has changed. Why? Due to the destruction of SH groups. Why SH groups of enzyme are destroyed, sir? Due to binding of non-competitive inhibitor. Where it has binded? Other site but not at active site. Okay, right? I am writing these statements. Okay, what they are, sir? What are the statements? Non-competitive inhibitor, non-competitive inhibitor, not structurally similar, not structurally similar to, it is not structurally similar to substrate molecule. Okay, is it structurally similar? Not structurally similar to substrate molecule. So, non-competitive inhibitor, does it competes? Does not competes. Does not competes with a substrate. Non-competitive inhibitor, right, it does not competes with a substrate. Non-competitive inhibitor, non-competitive inhibitor binds at what? Other site. This statement is very, very important. It's not binding at active site, it is binding at other site. Then what happens when it binds at active site? When it binds at other site, what happens? SH groups of enzyme destroyed. What are destroyed? SH groups of enzymes gets destroyed. Globular structure of enzyme changes. Globular structure of enzyme changes. Globular structure of enzyme changes. Sir, once globular structure changes, what happens? Shape of active site changes. Shape of what, sir? Active site changes. Shape of active site changes. So, write enzyme activity inhibited. Enzyme activity inhibited, enzyme activity inhibited, enzyme activity inhibited. Okay, right? Sir, we are discussing non-competitive irreversible inhibition. Yes or no? Sir, for non-competitive inhibitors, first you tell me where these non-competitive inhibitors are binding. Other side but not at active site. Other side but not at active site. What are the examples for non-competitive inhibitors, sir? Copper, mercury, silver, cyanide. What, sir? Cyanide. Okay, right? These are heavy metals, no? Right, okay. Now listen here. What is this one, sir? Cyanide, right? Cyanide poisoning of cytochrome oxidase. Cyanide poisoning. It means cyanide, it is inhibiting which enzyme? Cytochrome oxidase enzyme. Therefore, cyanide poisoning. Cyanide poisoning of what? Cytochrome oxidase. This is also an example for what, sir? Non-competitive irreversible inhibition. What is the enzyme name? Cytochrome oxidase. Who is inhibiting the activity of cytochrome oxidase? Cyanide. So cyanide poisoning of cytochrome oxidase is also an example for non-competitive irreversible inhibition, right? Go for the third one. What's sir? Non-competitive reversible inhibition. Reversible inhibition. Non-competitive reversible inhibition. See here. Sir, some enzymes, not all. Some enzymes, they contains allosteric sites. Some enzymes contains what, sir? Allosteric sites. Allosteric sites. Sir, what are these allosteric sites, sir? What are allosteric sites, sir? Sir, these are specific sites of an enzyme other than active sites. They are not active sites, but they are specific sites. They are not active sites, but they are specific sites. Okay. Therefore, some enzymes, they are with what, sir? Allosteric sites. Sir, which enzyme is having allosteric sites? The enzyme which is having what, sir? Allosteric sites. That enzyme is called as allosteric enzyme. What is that enzyme called as a allosteric enzyme? Therefore, enzyme with allosteric site. What is that enzyme called as a allosteric enzyme? What are the examples of allosteric enzyme? Number one, hexokinase. What is the enzyme name? Hexokinase. Number two, phosphofructokinase. Third one is a pyruvic kinase. What it is, sir? Pyruvic kinase. Therefore, hexokinase, phosphofructokinase, pyruvic kinase. These three enzymes are allosteric enzymes. It means they contains allosteric sites. Sir, these three enzymes, they are involved in glycolysis process. These three enzymes are involved in which step of aerobic respiration? Okay, which step of respiration? Glycolysis. These three enzymes are involved in glycolysis step of respiration. Okay, now listen here. Sir, enzyme with which sites are allosteric sites? Such enzymes are called as allosteric enzymes. Sir, take a chemical substance. That chemical substance binded in the active, that chemical substance binded in the allosteric site of an allosteric enzyme. Okay, if that chemical substance when binded in the allosteric site of an allosteric enzyme, if that chemical substance brings a change in the allosteric enzyme, then that chemical substance is called as allosteric modulator. Therefore, chemical substances, okay, which when binds in the allosteric site of an enzyme, if that chemical substance brings a change, brings a modification in an enzyme, that chemical substances are called as allosteric modulators. What they are called as? Allosteric modulators. Therefore, chemical substances What are chemical substances? Chemical substances which brings what? Modifications, which brings modifications in allosteric enzymes when these chemical substances, when this chemical substances binds at allosteric site. See this statement, sir, these are chemical substances. What they are doing? They are bringing some changes, modifications in allosteric enzymes. When they will modify the enzymes, sir, when these chemical substances binds at allosteric site, these chemical substances are called as what, sir? Allosteric modulators. Allosteric modulators. What they are called as? Allosteric modulators. Sir, the mechanism by which they are bringing modifications in allosteric enzymes, that mechanism is called as allosteric modulation. What is that mechanism called as? Allosteric modulation. The mechanism, right, by which a allosteric modulator, right, brings what? Brings modifications in an allosteric enzyme. Sir, by which mechanism a allosteric modulator is bringing changes or modifications in allosteric enzyme? That mechanism is called as what, sir? Allosteric modulation. What it is called as? Allosteric modulation. Sir, these allosteric modulations are of two types. Allosteric modulations are of how many types, sir? Two types. Allosteric modulations are of two types. Positive allosteric modulations. Second one is a negative allosteric modulations. Positive allosteric modulations, negative allosteric modulations. Sir, positive allosteric modulations means when a allosteric modulator binds in the allosteric site of an enzyme, then enzyme gets activated. Sir, negative allosteric modulations means when a allosteric modulator binds in the allosteric site of an enzyme, it inhibits the enzyme activity. Then it becomes what, sir? Negative allosteric modulation. Positive allosteric modulation means, right, when a allosteric modulator binds in the what? Allosteric site of an enzyme, the enzyme gets activated. So this what? So this positive allosteric modulations are done by activators or inhibitors? Activators. Sir, negative allosteric modulation. When allosteric modulator binds in the allosteric site of an enzyme, they inhibits the enzyme activity. Therefore, negative allosteric modulations are done by, are brought about by whom, sir? Inhibitors. Therefore, positive allosteric modulations by activators, negative allosteric modulations by whom, sir? Inhibitors. By inhibitors, right? 1 minute. For example, you take a phosphofructokinase. Sir, phosphofructokinase, this is a what, sir? Allosteric enzyme. It means it is having allosteric site. Sir, at this allosteric site, if AMP and ADP, any one of these two binds, then enzyme gets activated. Therefore, AMP and ADP, these two are activators. Which activators? Allosteric activators of phosphofructokinase. Sir, when ATP binds in the allosteric site of phosphofructokinase, then it inhibits the enzyme. Therefore, it is the inhibitor. What it is, sir? Inhibitor. Therefore, sir, phosphofructokinase is a allosteric enzyme. So, it is having allosteric site. In the allosteric site of this enzyme, either AMP or if ADP binds, the enzyme gets activated. Instead of AMP or ADP, if ATP binds in the allosteric site of this enzyme, the enzyme gets inhibited. Okay, right? Thank you.